Okay, I'd like to add my thanks to everybody else's for the organizers for bringing us together. It's great to see old friends and to meet some new ones. So I'm going to talk to you today about voltage imaging and some of the challenges associated with this. Today I'm not going to focus much on the biology. I'm going to focus more on the data aspects in the spirit of this meeting. And because of the limited time, I've summarized my talk in haiku form as follows. So voltage imaging, those movies are beautiful. Yikes, too much data. Okay, so now I'll go into a little more detail. The brain has interesting dynamics across a wide range of scales of space and time. And part of the fun and challenge of developing tools for studying brain dynamics is that there is no single tool which will capture all of these dynamics. And so we're working on different tools in different parts of this parameter space. Today, I'll focus on some tools for imaging subcellular voltage dynamics with very high resolution in space and time. This is real data from a CA1 pyramidal cell in an acute slice. And then also for zooming out and looking at circuit dynamics. Much fewer neurons than Ali Pasha is looking at. We're looking here at 300 neurons, but we're recording the voltage at kilohertz frame rates. And so we can see all the spikes and all the subthreshold voltages. We're also working on tools for mapping slower dynamics in the brain associated with plasticity at the level of individual spines, as well as in gene expression across the whole brain. But I won't talk about those today. Okay. So our interest in imaging electrical activity started with a more or less chance encounter about a decade ago with a molecule called arcuidopsin 3, which is a transmembrane protein which happens to be a little bit fluorescent. You can excite it with red light, and it emits in the near infrared. And the fluorescence of this protein is very sensitive to the membrane voltage. So here's a HEK cell expressing this protein, and we're using a patch pipette to move the voltage up and down. And as the voltage goes up, it gets brighter, and as the voltage goes down, it gets dimmer. And we realize this gives us now a window into seeing bioelectrical phenomena. Now, here we're using a patch pipette to trigger this electrical activity in the cell, and that partially defeats the purpose of having an optical readout because you still have to go in and poke the cells one at a time to stimulate them. These voltage indicators are excited by red light, and they emit in the near infrared, and so that leaves the rest of the visible spectrum open for other uses. For example, we combined the voltage indicators with some blue light activated channel rhodopsins so that we can stimulate the neurons with blue light and record from them with red light. And there's a cartoon of that, and this is some data from, this old data from a cultured neuron, and you can see each time we deliver a blue flash to the cell, you can see a flash of fluorescence, and here's a patch clamp recording taken at the same time just for ground truth. But now we have a neuro-optical interface, so we can stimulate and record with light. And part of the charm of having that is that you can now image electrical activity over large fields of view. This is an example of cultured neurons. There's about 700 neurons in this field of view. They've been pseudocolored just so we can tell them apart. And here I'll show you a movie taken at 1,000 frames a second, slowed down about 50-fold, where at a certain point we'll stimulate all the cells and you can see them start to fire away. And we can segment these images and extract the spikes and all the subthreshold voltages of all the cells in the culture. Okay? And so that's become a tool which is now being used for a variety of disease modeling and drug discovery applications, which I'm not going to get into. Today, I'm going to talk about our efforts to use these tools in intact tissue to watch real neural circuits. And so we've developed optical systems which combine patterned blue light off of a digital micromirror device for targeted optogenetic stimulation, and patterned orange or red light off of another micromirror device, which can be used for structured illumination. You only want to put photons into the sample in places of parts of the sample that will generate useful signal, and you don't want to put any photons into the interstitial regions. This is all one photon imaging, and so we're very sensitive to background issues. And then we also have a 2-photon microscope, which we can use for structural imaging. We'll use a blue light activated channel rhodopsin, and the voltage indicator we use is called Voltron 2, which is a more advanced version of the voltage indicators that I showed you at the beginning. This was developed by Ahmed Abdel Fattah and Eric Schreider at Janelia Farm, and it uses a fluorescent tag attached to the opsin to amplify, to make brighter the voltage-sensitive signal. And this is what the setup looks like when it's running, so it looks sort of like an atomic physics experiment, except there's a mouse somewhere in there instead of cold rubidium. Okay, so first I'll give you a vignette about using this to image subcellular voltages. So in my conception, a neuron is a device that takes inputs at its synapses and it makes spikes, and then those spikes go down the axon and they go back up the dendritic tree where they can interact with synaptic inputs to drive plasticity. We'd like to know what is the input-output of the function of the neuron, so what's the relation between the patterns of inputs and the patterns of spikes. There are many voltage-gated channels in these dendrites, which make this into a rather complex nonlinear function. And we'd like to know the plasticity rules. So how does the pattern of inputs and the pattern of spikes lead to changes in the input-output function of the neuron? This is simplistically summarized by the notion that neurons that fire together wire together, but simple Hebbian plasticity is not dynamically stable. You'll get runaway plasticity where the synapses that get stronger become more correlated, which then makes them stronger and so on. And so you need some more complex plasticity rules. Now, over the last 25 years or so, many people have done beautiful patch clamp experiments which have identified a zoo of different nonlinear excitations which can occur within dendrites. And so there's calcium spikes and sodium spikes and NMDA spikes. Our goal in this effort is to try to relate the biophysics of these different kinds of excitations to some understanding of the computational role of the neuron. Okay? And this is a long story, and so I'm not going to go through all the details here. I'm just going to give you a flavor. So in acute slices, we can use our video projector to target the blue light to different parts of the cell and to map the response. Here's an example where we're targeting the cell body, and then the raw video are taken at 1,000 frames a second, so 1 millisecond a frame. But because we know that the underlying action potential is a smooth function of time, which goes up and comes back down, we can interpolate these discretely sampled data to increase the time resolution and to map the wavefront with a precision much better than the exposure time of the camera. And so here's an example where we've interpolated the wavefront to about 50,000 frames a second. And here's the map of this action potential back propagating throughout this dendritic tree. This is based on an average over about 100 trials. Okay. Now, we can take these neurons and we can stimulate different noodles. So here the blue here shows where we're stimulating. And we can then map the patterns of electrical response in the cell. And things start to get sort of weird. So here's an example where we're stimulating this noodle here. The orange shows the voltage at the cell body, and you can see the neuron starts to spike away. But if you look out in this distal dendrite, you can see the first spike doesn't make it, and then a couple of spikes do make it, and then a whole bunch of spikes don't make it. Okay? So there's some very strange history-dependent nonlinear amplification of these signals as they go out into the dendrites. And this is a robust effect over many cells. Depending upon where we stimulate them, the details change. But basically, over many cells, a stepwise increase in the spike rate at the soma leads to, after a little delay, a transient increase in the amplitude of the voltages out in the dendrites. Okay? And so we can summarize all of these data by saying that the dendrites look like they're acting like a spike rate accelerometer. So when you have an increase in the spike rate at the soma, you get a transient window where those spikes can make it into the dendrites. And we think this has important implications for plasticity rules, which I'd be happy to discuss maybe during a break or some other time. Now, these experiments also let us map the spatial footprints of the voltage within these cells. So here is an example of 5 views of the same neuron. The blue shows where we're tickling the neuron. In the middle here, I show the maps of the subthreshold voltages evoked by these different stimuli. And you can see wherever we stimulate, the depolarization is largest, and then the influence of that stimulus decays as you move away from the stimulus location. And that's not so surprising. Then the question we asked is, does the spatial profile of that back-propagating action potential know anything about where the stimulus came from? Do these things know where they were born? And you might think that the answer would be yes, because there has to be some information about where that stimulus came from in order for plasticity eventually to be localized to the location of the stimulus. To our great surprise, we found that the answer was no, to the best of our ability to measure it. So both back-propagating action potentials that do not make those dendritic spikes, as well as the ones that do make the dendritic spikes, have quite stereotyped spatial footprints which do not contain information, as best we can tell, about the location of the stimulus that evoked them. And this was a surprise. This suggests to us that the information about where the stimulus came from must be stored in some chemical degree of freedom as opposed to an electrical one. And there's a whole story about dissecting that. We think this has to do ultimately with the gating states of NMDA receptors, and that's where the spatial information is stored. Okay, so here we have these data where we have large data sets on single cells, and we can use these tools to stimulate them in different patterns of space and time to try to map their input-output functions. Now I'm going to tell you about another flavor of data which the same basic tools can provide. So we're going to zoom out now and use a large objective lens to look across as much of the brain as we can see. And because this is one photon imaging, we're limited to relatively superficial regions of tissue. So here's our field of view. It's about 3 millimeters across. And we're looking in layer 1 of the cortex, because that's what we can see most easily. So we're looking at the layer 1 interneurons. These are inhibitory neurons, which form a sparse mesh sitting over the top of the cortex. And we express the channel rhodopsin and the voltage indicator in them. We use the video projector to flicker the individual pixels on and off in some unique patterns which let us assign the fluorescence signal from each spot in the sample to it's basically a digital confocal. So we can turn on and off individual pixels in the projection and use that to map the three-dimensional structure of the tissue. We find the cells, and then we use the video projector to only target the illumination to the cells we want to record from. Okay? And so this way, we avoid putting light into all of these interstitial regions, and you can see you get a big improvement in signal-to-background ratio just by restricting the illumination. And so here's what the data look like. Here we're recording from about 300 neurons. This is in an awake mouse, just spontaneous activity. Here's the time scale up here, and you can see every spike and all the subthreshold voltages in all of these cells. We can also go in and stimulate the cells in different patterns of space and time. Here's an example where we turn on a stimulus, broad stimulus, partway through the recording, and you can see that the cells start to spike more when we stimulate them. And we do these experiments in awake mice, which are on a treadmill, and so they can run on the treadmill, and we can record their running, which is shown here in red. We also take video of their faces, and so we can look at their whisking and their facial motion and their pupil diameter. We can stimulate their whiskers if we want. And then we can record from all the neurons. Here we've set aside the subthreshold voltages, and we're just looking at the spikes. And so each row here is one cell, and each point is one spike. And then we can also go in and stimulate. And so here's an optical stimulus pattern, which is targeted just to this subset of neurons up here, and you can see that those spike a lot more when we stimulate them. And so we now have the ability to see basically everything that these neurons are doing electrically and to perturb however we want. And so here's an example of how we're starting to explore this circuit. We pick a sparse subset of the cells, and we stimulate them, and then we adjust which subset we're stimulating periodically. And by triggering the activity of all the other cells off of the neurons that we're stimulating, we can map the synaptic connections between each cell and every other cell, as well as the gap junction connections. We have the complete functional connectivity matrix between every pair of cells in the circuit. And then we can look at how this circuit responds to, during spontaneous behavior or during sensory inputs. And that's sort of where we're at now. We have many terabytes of data of this sort, and we're trying to make sense of it. And we've been working on that now for about a year and almost 2 years. And the data acquisition takes maybe a day or two, and the analysis has been the work of several years. I want to say a little bit about the software which is supporting all of this effort. These experiments are rather complex to design because you have to pick which cells you're going to illuminate and which ones you're going to stimulate. And so we've developed a somewhat user-friendly software package for controlling these experiments. And we haven't really disseminated it broadly yet, but we've set up a little website for it. And if you're interested in doing these kinds of experiments, I'm happy to discuss. It turns out the instrument for this is actually not that complicated. For these experiments, we're using just an LED to illuminate the samples and some video projectors and commercially available lenses. The other side of the computational problem is on the analysis. Here's a summary of the pre-processing pipeline. So we get about 1 gigabyte of data per second as these run, so a couple of terabytes per day. And then we have to denoise it. There's a variety of sources of both systematic and statistical noise. We have to pull out the signals from the individual cells, and then we can get these traces. And then that is sort of where the science begins in terms of trying to understand what drives these different patterns of activity. So I want to end by talking about some of the unsolved challenges, which I hope people with more computational expertise than I have can help us with. So we're drowning in data, and we are looking for help developing better signal extraction and data distillation algorithms just to get us down to the traces. And then the really interesting problems are around dealing with these high-dimensional data. Both we have the behavior as well as the neural activity, and the ability to stimulate and perturb this neural activity in real time. And so ultimately, we're hoping to start closing the loop to look at the patterns of activity and to use that to inform which neurons we stimulate or silence. And then the last thing I'll mention here is this question of how do we share these data. This is really intractable at the moment, and it's been very difficult for us to comply with the NIH mandates in any sort of meaningful way. And I hope this is a topic that we can discuss, because I think there's a lot of energy being expended for unclear scientific value. So with that, I'd like to thank the people who did the work. The work I showed you today was in dendrites, was largely driven by David Wong Campos and Pojung Park, both of whom are on the job market, so you should hire them, please. As well as the network activity was done by Yutong Chi, who is a grad student in my lab. This is my lab, and I'm looking for new people to join us, and I'm happy to take questions. Thank you.
Yeah, question?
Yes. Okay.
Yeah, so first of all, beautiful talk.
Thank you
So much. I'm curious where you think the limitations are in applying this technique to imaging in depth.
Sure.
And if can I just add one more thing? If the limitation is in the fact that you need to go at 10,000 hertz, would there be a way perhaps to multiplex? So you do voltage imaging at 100 hertz, because nothing's going to happen above that, except for spikes, and then some other form of imaging to get the spikes.
Yeah, so it's of course a multi-part problem. It's all one photon imaging, and so ultimately the challenge we face as we go deeper is light scattering. So as you go deeper, you get more and more light scatter, and eventually the signals just get lost in the haze. Now, you can partially mitigate that by going further into the red part of the spectrum, where you get less autofluorescence and less light scatter. And so there the challenge is a molecular one of developing good, bright voltage indicators which are further to the red. You can partially mitigate it with structured illumination and with adaptive optics, so you can partially compensate for some of the distortions, aberrations induced by the tissue. But those help somewhat, but they don't really, they're not going to get you as deep as 2-photon imaging. The other things we've done, so for instance, if you're interested in looking at the hippocampus, you can scoop off the cortex and then you can look in hippocampus. And we've been doing that, looking at wide area dynamics in the hippocampus as well. And then we've been implanting prisms. So the prism can give you a side-on view through a piece of cortex. To get really deep, ultimately, two-photon microscopy has the capability to get deeper, but it's very difficult to get enough photons out at a high enough rate to overcome the shot noise with two-photon microscopy. And there are many other technical challenges there as well. We're working on it, but I think getting wide area, two-photon recordings from many neurons with voltage is, I have no idea how one would approach that. I think hybrid approaches using calcium to guide you and then voltage to zoom in or to pick subsets of cells or to multiplex, I think could be very powerful. Yeah.
Okay, thank you. I'm sorry, we do have to move on, but we have a discussion panel afterward. Thank you so much.